In the previous module, we saw the ultrastructure of microtubule, how different subunits are put together, how the subunits of microtubules are put together to form this long cylindrical structure, microtubule. I would like to again talk about the polarity, but now I'm going to talk about polarity in context of actin filaments. We will see the subunits, how these subunits of actin monomers, they come together and form the filaments, and what makes them polar structures, meaning the two ends are different. The actin subunit is a single globular polypeptide chain and is thus the monomer rather than the dimer. In case of microtubules, we saw there were two polypeptide chains, alpha and beta. They came together and formed the subunit. In case of actin, it's only a single polypeptide. Each actin subunit either binds to an ATP or an ADP. We saw in case of microtubules, it was the GTP or the GDP. We'll talk about the differences later on. But actin filaments, they bind ATP rather than GTP. Actin subunits assemble head to tail to generate filaments with distinct structural polarity. I'll show you the diagram later, but for now, I would like to say that in cells, actin filaments are cross-linked and bundled together by a variety of accessory proteins, making the large-scale actin structures much more stronger than the individual actin filaments. So strength in numbers, so just when you bundle up linear fibers and you tie them up, you can increase the strength of the resulting fiber. Just like ropes you have, if you look at a rope, it is made up of individual tiny thin strings, but bunch of them together, they can form a much stronger structure altogether. So now let's look at the structure of actin monomers. Here is the actin monomer. The red part here is basically the yellow part, is the ATP bound to the monomer. I'll highlight it here. This is the ATP. And this is basically the minus end of actin monomer, and that is the plus end. So addition, when the new monomers are added, they preferably be added at the plus end, and at the minus end is basically where the monomers are lost in the actin filaments. So these monomers, they come together and form actin filaments. If you look at this filament, actin filament, right here, you will notice that it also has some sort of a helical structure. If you think about it, it seems as if there are two filaments coming together and wrapping around each other, which is repeated. This basically narrowing or broadening of the actin filament, it repeats every 37 nanometers, and that is because they are also forming a helical structure. So here you have the plus end of the actin filament, and here you have the minus end. It is basically how this monomer is placed in the three-dimensional space. This part is basically, this part of this globular protein is exposed in the minus end, where at the plus end, it is this part of this globular protein which is exposed the other end of this filament. Here you can see the actin electron micrograph of actin filaments. So this is basically the important points that I wanted to make on this diagram. Let's move on to the next slide. The actin filament can also, as you can see, as I mentioned, can be considered consisting of two parallel protofilaments that twist and wrap around each other in a right-handed helix. Actin filaments are relatively flexible as compared to hollow cylindrical microtubules. I would like to point out, in case you are looking at literature and you find different terminology, for historical reasons, the plus end of actin filaments is also referred to as the barbed end. So you may come across literature in which they're talking about barbed end of actin filaments. That is basically the plus end. And the minus end is referred to as pointed end because of the arrowhead appearance of myosin heads when they are bound along a filament. So the plus end, the minus end the barbed end and also the pointed end. So this is basically the ultrastructure of actin filament, and as we will see when we talk about cytoskeleton further, the result, the consequences of the two ends of these filaments being different and how this different manifests itself along the length of these polymers.